We're going through biology, excretion and homeostasis. So it's just a revision of what you learned in class. So one, you must know the meaning of excretion and also the meaning of homeostasis. So excretion, it involves separation and elimination of waste products of metabolism from the living cells. It is mainly done to avoid poisoning or poisoning the respiratory cells within our bodies. When it comes to homeostasis, we are maintaining internal environment despite changes in external environment, or internal temperature despite changes in external environment. The excretion first, and in biology we have the botany part and the zoology part, and in most cases we start with the botany part, which is the plant part, and then we move to zoology in plants. We have so many things we look at. Number one, we look at why plants lack why plants lack why plants lack complex excretory organs. Why plants lack organs. We underline the word lack. Why they lack these complex excretory organs. We have reasons why plants lack. But when we'll be looking at animals, literally they have complex excretory organs. So the reason why plants lack, one of them, the metabolic activities in plants are few. So, or we can say the metabolic activities are few and they produce or release metabolic wastes slowly. So, most of the plants, they normally release or they produce these metabolic activities slowly. So we have releasing or producing metabolic wastes slowly. Toa those wastes pole pole. Unlike animals, zinakuja zinaitoa mostly at once. So utapata zile zikitoka haraka, or when they are produced faster, accumulation inakuwa very high, so they might cause intense effects. But when you look at plants, zinaitoa pole pole. The small quantity have less effect on plants. So also plants produce less toxic waste because mainly they break down carbohydrates to produce carbon dioxide and water. When you look at the process of photosynthesis, we have look at the process of photosynthesis. We have CO2 plus water. You will get C6H12O6 plus oxygen gas. So here we have chlorophyll molecule, and then we have sunlight, or you can say light energy. So we are having the raw materials, then we are having, these are the conditions necessary for photosynthesis, and then we have, these are the byproducts. byproducts. So we will find that we are having carbon dioxide gas and water, which are raw material for this to take place, in presence of light energy or the sunlight and chlorophyll molecule. We will have a carbohydrate. In most cases, this is glucose. Glucose, it is produced fast. Then normally this glucose is normally converted to starch. Why? Because glucose is normally osmotically active, and we have reasons for that. Why glucose is not stored in plants. So it will be converted to starch. Starch is osmotically inactive, so the starch is the form of carbohydrate which is stored in plants. And then we have plus oxygen. So we'll find that these are the raw materials. So we'll find that we've said some within the plant. For example, the carbon dioxide and water are used for photosynthesis, and some oxygen is used for respiration. So this is a byproduct. This oxygen is supposed to be released to the environment. But what happens is this: when oxygen is produced, which is a byproduct, or we can say is a waste, the oxygen in the process of respiration, but excess oxygen is released to the environment. That's why you'll find people say plants give out CO2 that is produced. That is oxygen gas, is the excess oxygen. But the one that is produced is used in the respiration process. So the excess is released to the environment. So we have most products are converted into non-toxic or harmful substances that are produced into the this oxygen we've talked about, which is a byproduct as a result of photosynthesis. But the same oxygen is used by the same plant, but that excess is the one that is released to the environment. But it's not that in fact, oxygen is not toxic. It is, of course, it is utilized by living organisms. The zoology part, that is the living the animals. So plants wastes are stored in temporal structures in less toxic form, which then fall off. Like for example, the fruits and the bark. So some of the waste products in plants, they are stored in leaves. At some point, you'll find some leaves, they turn their brown or somehow a yellow. Some of the waste products are stored in flowers. After the reproduction process, that is the double fertilization process, the petal part, it withers off. When it withers off, and remember, the waste products are stored inside those flowers. So when it withers off, it goes with the waste products. Same to leaves. When these waste products are taken to the leaves for storage, then these leaves falls off. What will happen? It's falling with the waste products in it. So that's how it is eliminated. The barks of trees, that's the old phloem tissues. You'll when you visit the forest, or even at home, you'll find that we have the barks of trees. They normally follow gradually. So these waste products, when they are stored, they're stored. For example, this is a tree. So the outer part, inner part, that is the xylem. Let's assume this is the bark of tree. This is the bark. Let's say this phloem, you can say the bark of tree. Then when these waste products come, they accumulate at the phloem tissue of the bark of. Then at some point, when the growth and development takes place, the bark of trees, they start or they have gradual falling off. Remember, the waste products have been stored at the back of trees. So when these backs of the trees are falling off, so they will fall off with the waste products. Those are crucial complex story organs. So at these points, you should know them at your fingertips. So we also have methods, uh, methods of excretion in plants, or the process of excretion in plants, or how does excretion in plants occur. So we have excrete. Number one, we have the diffusion, we have the transpiration, we have the deposition, we have the tissue organ fall off, of which we have discussed. We have exudation, and we have the guttation. We start with the guttation. Excess water is excreted in form of droplets through the hydatodes, and mostly this one happens in most cases at night. These are excreted in semi-solid form. At some point, when you have the Gravilla robusta trees, then it is in backs of trees. So you'll find that we have some thick liquid that comes out. That's an example of a exudation. And then we have tissue organ fall off. That's we said about the leaf fall. Let's say the back of trees when they fall off. So when the waste products accumulate, and then in the leaf, let's say a leaf or even the flowers, and then the flowers falls off, it has got in an untoxic form. So some waste like calcium oxide, caffeine, nicotine, and quinine are stored in tissues, or that's the leaves, the fruits, flowers, and all those waste products. Most of them are non-toxic, so they are less toxic to the plant. Here we are talking the effect of. So you'll find that most of these waste products are non-toxic in plants. So and we have diffusion. For example, the carbon dioxide from respiration and oxygen from photosynthesis are emulated through diffusion through the lenticized cuticle and stomata. And remember, on diffusion, where these gases are highly concentrated, they tend to move to where they are lowly concentrated. So you'll find that when carbon dioxide is highly concentrated in the respiratory cell, so what happen? It will now diffuse, and then you have the when the oxygen, let's say oxygen is more oxygen is highly concentrated in leaf cells. So what will happen? It is highly concentrated in leaf cells, is lowly concentrated outside the leaf, so it will diffuse out. And then we have the transpiration. Excess water is excreted in form of water vapor. Have the cuticular and have the lenticular transpiration. So those are a brief introduction about it. And then we have economic importance of plant. Economic importance, that one we won't go through them. We will look at those. We have to have some serious discussion on homeostasis, because here is where most of the problems comes in the homeostasis. So at some point somebody may ask, eh, homeostasis, we had talked homeostasis is a mechanism, environment, despite or regardless of the external conditions. That is homeostasis. So when you look at homeostasis, we have different types of different effects of homeostasis. For example, somebody may ask, just maintained constant in mammalian bodies. Factors that must be maintained constant in mammalian bodies. factors that must be maintained constant in mammalian bodies. We have temperature, water, salt or iron content, CO2, have the glucose, have the amino acids. These are some of the factors. You can say temperature must be role of liver, role of skin. You'll find that temperature, in most cases, temperature has a great effect, especially in living cells, where temperature affects how or how we talk of the membrane. Remember, the cell membrane is semi-permeable, and semi-permeable means we have materials passing through, but materials of large sizes are not allowed to pass through. That is semi-permeable, and that is one of the characteristics of the cell membrane. We have the cell wall. Cell wall, it is material of different sizes to pass through. Now, when you look at the especially in animals, now we are talking of homeostasis in animals. When you look at the cell membrane in animals, which is semi-permeable, materials of small sizes pass through. Remember, in semi in have we have what we call a lipoprotein layer. We have the phospholipid layers. So high temperature and also acidic conditions, or the pH. The pH. So we've not mentioned pH, but we're just talking the effect of temperature and pH. So we'll find that those temperature or to extreme pH, when it is the spore, remember the pores are too small, they're minute. So when they are subjected to high temperature, may affect their size. Either it may close completely or it may tend to open. So when it closes, even those small materials will not enter. And if it opens wider, it now becomes almost like fully permeable. Materials that that are not supposed to enter, they'll find their way into the cell membrane. So what will happen? We will have dysfunction or malfunctioning of the cell membrane. Now, we have a question here. Outline and explain. Outline and explain. Outline and explain homeostatic functions of the liver. Outline and explain various homeostatic functions of the liver. Now, liver has different functions. We normally talk of the two main functions. One of them being the deamination and deamination. Deamination of removal of an amino group. So removal of amino group removal of amino group from the amino acid molecule. From amino acid molecule. So amino group inatolewa from the amino acid. So the process gets rid of excess amino acids in the body. Literally here, the amino group inside the amino acid, we are assuming we have the excess amino acid in the in the cell or in the body. So this amino group is eliminated or is removed from the amino acid molecule. So the process gets rid of excess amino acids in the body as the body is not able to store them. And we are talking of at some point excess. So if something is excess, cannot be stored, more of it cannot be stored, so the excess is removed, and that is the amino group which is removed from the amino acid molecule. So you'll find that the body is not able to store them, and the amino group enters the ornithine cycle, where it is combined with the carbon 4 oxide from the urea, and which is excreted in the urine through the kidney. Literally, deamination is just removal of the amino acid group from the amino acid. All these are processes just explaining how the amino acid is produced. So heat production, that is number, we have heat deamination, and then we have the heat production. And heat production, many metabolic activities take place in the liver. Inside the liver, we have so many metabolic activities. Inside the liver. So we find that release. Okay, inside this liver, we have release of heat energy that is distributed by the blood to other parts of the body. Remember, blood. We have blood has a various function. Number one, you have thermoregulation. Thermo comes from heat, so it regulates it. In fact, at some point, you say it ensures blood ensures we have the equal distribution of the heat inside the body. In a move now that heat, that's why you'll find that huwezi pata mkono it is warmer, mguu it is cold, let's say kidole is warm. No, so we have the blood that ensures we have equal distribution of heat inside the body. And then we have the kidney, which releases the energy that is distributed by the blood to other parts of the body, and this helps in thermoregulation. We also have storage of vitamins and mineral salts. We have storage of vitamins and mineral salts, D, E, and K. So when you talk of vitamins, we have A, have vitamins, have vitamin A, B, D, E, and K. This is E and K are stored in the liver. So these are stored in the liver. So they are stored in the liver, and worn out red blood cells are broken down to yield the iron. The worn out red blood cells are broken down to yield iron, which is stored in the liver in the which is stored it is stored in the liver in the form of in the form of stored in the liver in form of ferritin, that is it is used later in the case of shortage. So this one is stored, iron is stored in form of ferritin. Now when the body has shortage of iron, this ferritin is now used back. It's going to be used back because that's just a storage, storage. So we have uh-huh we have also the formation of red blood cells, which occurs in there, and we have the liver also breaks down old or exhausted red blood cells, leading to formation of the more in the bone marrow to replace the worn out cells. Remember, this bone marrow contributes to blood formation, where we are seeing the formation of the red blood cells, which is happening in the bone marrow. And then we have it enhances oxygen and carbon dioxide distribution. Remember, oxygen and carbon dioxide distributed inside the blood. So when we talk about gaseous exchange, go back to gaseous exchange. When you breathe in oxygen, the nose, inside the nose, it is moist. All the respiratory surface, not leave alone even the nose, all the respiratory surfaces are moist to dissolve the gases. Those gases, they go and dissolve in the blood, is transported in the blood. So you'll find that oxygen will combine with hemoglobin. Oxygen plus hemoglobin, you'll find now hemoglobin. This is blood rich in oxygen. You'll find carbon dioxide plus hemoglobin. You'll find carboxy hemoglobin, which is now rich in carbon dioxide. And remember, this oxyhemoglobin, it is an unstable compound which normally dissociates very fast, yani inayeyuka haraka sana. Carboxyhemoglobin is a stable compound that does not dissociate. That's why at some point when you breathe in let's say a gas rich in CO gas, that is carbon monoxide, it will combine with hemoglobin to form carboxyhemoglobin, which is a stable compound, and it will clot. The clot will be formed. So it will hinder the normal flow of blood. That's why at some point you find people suffocating because of that, because of this carboxyhemoglobin, which is now a stable compound that does not dissociate. It does not dissociate. Then let's continue. We have regulation of blood sugar level. Regulation of blood sugar level. Regulation of blood sugar level. We're still looking at the liver, homeostasis in the liver. Regulation of the blood sugar level, and have the liver cells convert excess glucose to glucogen. So we have glucose. In fact, it's excess. Excess glucose is converted to glucagon. Not glucagon, glucogen. Converted to glycogen. 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 So glucose, excess glucose it's converted to glycogen, and fats under the influence of insulin hormone and and fats, and whatever happens here have the insulin hormone that leads to conversion of excess glucose to glycogen and fats. So the stored glycogen is, however, converted back to glucose when the glucose levels are low. So let's write, when the glucose, we have glucose high and glucose low. So glucose, when it is high, it is converted to glycogen, and fats with the influence of insulin hormone. Then that is it. When the glucose is glycogen and fats, and that is under the insulin hormone. But when the stored glycogen is converted back to glucose when the levels are low. So when it's low, the glycogen is converted back to glucose. The stored glycogen is converted back to glucose when the levels are low. So by the liver cells under the influence of glycogen hormone. So we have the insulin hormone here, and then we have the glycogen, insulin hormone under high when the glucose is high, converted to it has stimulation of, glucose converted to glycogen. But when it is low, the glucagon hormone converts the glycogen to now glucose. Just a reverse. When glucose is high, it is when the glucose is low, it's converted to glycogen. That the stored glycogen is converted to glucose or back to glucose, but under the glucagon hormone. So regulation of plasma proteins, that's another one. That's about, we're on regulation of, have the sugar level. At some point we normally say the glucose level. So when you see sugar level, that's the same as glucose level. The excess glucose is converted to glycogen and fats under the influence of the insulin hormone. But when the glucose level is low, glycogen was converted, or the glucose was converted to like glycogen when the levels were high. But when it is low, the stored glycogen and converted back to glucose. That's all about the blood sugar, or the regulation of the blood sugar level. And so we are moving to regulation of plasma protein. Regulation of plasma protein. Regulation of plasma protein. Regulation of plasma protein. And plasma proteins such as the prothrombin and fibrinogen are informed too when we were learning about the blood clotting process. We encounter the prothrombin and the fibrinogen. So the fibrinogen and prothrombin are examples of the plasma proteins. So they are manufactured in the liver using the amino acids found in the liver. At some point, we said we have the amino acid that are found in the liver, but we have at some extent we process, which is the removal of the amino group. In the case of excess amino, that is from the amino molecule. So you'll find that when you're talking of the relation of blood plasma proteins or the plasma proteins, so we have the proteins found in the liver. So they play a major role in the blood clotting, as we've said, that is under the blood clotting system. So they prevent excessive blood loss and infection. And we have examples. We have the what prothrombin, prothrombin, and in blood clotting, in blood clotting. So when you wire mesh-like structure at the injured area, so the wire mesh-like structure prevents or stops the bleeding. So that is the controlling the excessive bleeding. Proteins. So other plasma proteins are produced by the liver. We have the serum and the albumin, which contribute to the maintenance of the osmotic pressure in the body, and the non-essential amino acids are also synthesized by the liver. We have the blood storage. Blood storage, highly vascularized. Highly vascularized means it is densely supplied. Is densely supplied with blood capillaries. I find it. We said blood storage. Say the liver is highly vascularized, and highly vascularized means it is densely supplied with blood capillaries. Densely supplied with blood. Iko blood capillaries mingi, it has more blood capillaries. So when we say it is highly vascularized, we mean it's highly vascularized. In other words, we can say highly supplied with blood capillaries. We mean that a lot of blood is more blood capillaries, which is responsible for warming the air. Remember, blood plays a role in thermoregulation. So, and it is capable of holding a large volume of blood when the blood vessels dilate. Remember, we have vasodilation, and we have, so dilation is like those capillaries are somehow increasing in size, zinaongeza, that is in diameter, zina increase. Vasoconstriction kana reduce in diameter, kanakuwa kadogo. So that is vasoconstriction and vasodilation. And here we are talking of the vasodilation. So volume of blood when the blood vessels dilate during hot conditions. Why do blood vessels dilate during hot conditions? To carry more blood. When it more heat, when more blood comes with more heat, so more heat, it comes to the almost this surface of the skin. More heat is lost to the environment, that is bringing about the homeostasis function. So you'll find vasodilation in an increase in size. With that heat or more heat inside the body, it carries with it when it reaches beneath the surface of the skin, so the heat is lost to the environment. It brings about the cooling influence of the endocrine and the nervous system. So kama temperature iko kidogo, We are talking about the internal temperature, not the external. When the internal temperature is low, what happens? Materials would be specific, so they vasoconstrict, kanarudi kanakuwa kidogo, so that it carries less blood, and less blood comes with less heat in the body. Less heat is lost. But when the internal temperature is high, vasodilation takes place, more blood passes through the capillaries. So we have the influence of the endocrine system and the nervous system. So less blood is stored in the liver when thermoregulation. When you talk of the blood storage, you talk of the thermoregulation. That is the regulation of the internal but this regulation, thermoregulation. Number two, we have the transport function. Number three, we have the blood clotting. Defense. So defense done by the white blood cells. Clotting is done by the platelet in the blood. Then it's transported. You have the thermoregulation, regulation of the body temperature, or you can say the equal distribution of the body of the temperature inside the body, done by the blood. Detoxification. 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 Process where harmful compounds such as drugs and poisons are converted to less toxic compounds in the liver. So you have conversion of harmful compounds like poisons to less harmful compounds, or to less, considered less harmful or to less toxic compounds. So urine, for example, we have some people who consume food. They consume food even without properly cleaning their hands. Has or contains a lot of germs, or contains a lot of harmful substances. You consume them, even some small, for example, let's poisonous content. So when it reaches the liver, the function of the liver is to make that poisonous content to be less poisonous. That's why we're saying the detoxification of harmful compounds, or the toxic compounds to less toxic compounds. So the toxic compounds are later excreted in the urine cells. Toxins in the body cells. So it does what? Prevents accumulation. Detoxification prevents accumulation of toxic toxins in the body cells. We all know the effect of toxic substances in the body cells. So detoxification prevents accumulation of toxins in the body cells with malfunctioning of the body cells. The body cell might malfunction. They might not function the way that is, they might not function properly, or death. That's about the broad discussion about the role of homeostasis in the liver in mammals. So, let's see how the another investigation discussion. We're going to look at how how endotherms respond and heat and cold conditions in their environment. How endotherms respond to heat and cold in the environment. Your body is subjected to extreme hot conditions or extreme low conditions. Now we are starting with hot, hot stroke heat conditions. So increase number one, when you have hot conditions or you have the heat conditions, we normally have increased sweating, and the increased sweating is made plants to experience transpiration, whereby how does it take place? We have the latent heat of vaporization, that takes place inside the leaf. The latent heat of vaporization, it makes water to change its form to water molecules, then escapes through the stomata. That is now transpiration, and that takes place in the leaf. But in human beings or in mammals, those heat or hot conditions, we have increase in sweating, and this sweating comes as a result of loss of latent heat of vaporization. And this latent heat of vaporization, it is intended to lose heat from our body. So when sweat comes out, it comes out with that. Kumbuka, sweat is a combination. Water is coming out. Of course, water will evaporate, and we have those salts. We have some those amino acids. The combination of amino acids on your skin. They will be seen as strips on your skin. We'll see some strips when somebody has engaged himself on strenuous activity. Then in terms those sweat glands, they produce the sweat, which sweat travels through the sweat duct to the sweat pore. Kumbuka here that sweat is internal temperature is high, so it's coming with that heat. When it reaches the surface of the skin, water will evaporate. When water will move, saying hot condition increased sweating to lose heat. said we have one dilation, the arterioles, whereby when the arterioles dilate, blood passing under the skin, more it carries more heat, and more heat will escape or will be lost to the atmosphere. And then we've said about the decreased body metabolism, that is still under heat, have decreased body metabolism. So decreased water metabolism the concept behind it is to reduce body reduce body metabolism and produce heat generation. That's reducing generation, reducing heat generation. And we have now the rectopili muscles relax. That's we are now talking about the skin. Rectopili muscles relax, making the hair follicle to relax, hence the hair light starts at no rectopili muscles. So what happens when you have the hair follicles relax? These hair follicles now they relax. They relax. When they relaxes, the hair now lies. After lying on the skin surface, this is the hair that has lied on the skin surface. Now, what happens? No is trapped, or at some point we say less air up. Remember, air is a poor conductor. When less air is trapped, it means that there is no mechanism in which the heat from the body will be lost. So it will just be lost because air has not been trapped. So the heat, excess heat, will be lost in the environment. But when it is cold, you'll find that the hairs will stand still or will stand erect. Air is a poor conductor of heat, so it will prevent heat loss from the body surface. It will prevent the heat loss. To lose heat, reduced muscular activities to have also due to slow metabolism, and also to reduce heat production, to expose tongue and mouth, mostly experienced by the dogs, have panting and exposing of the tongue. Avoid direct heat. That's a estivation. Have some animals they tend to estivate to escape the extreme heat. For example, we make hot temperature. We have the flapping of ears, create current to carry away heat, and have cold conditions. Stamping of feet heat directly, and have less production of sweat, reduce water loss through the latent heat of vaporization. We have the vasoconstriction of arterioles, hence less blood flow to the skin surface, reduce heat loss, and contract. Cooling hair follicles, hence hair is raised, and to trap a layer of moist air to prevent heat loss. And we have the rapid construction of the muscles to yield heat to warm the body. And so that is an easy question of 20 marks. So when you go through it slowly, the cold condition is the opposite of the hot condition. You have happens under hot conditions. We move to cold conditions. Cold condition is just hot conditions. For example, in hot condition you have dilation of the artery. In cold condition you have now constriction of arteries. You have decreased body metabolism. Under cold condition you have increased body mechanism. Under hot conditions we have irritability muscles relaxing. Under cold condition, now is the opposite of the hot condition. That is it about the end of we'll send them to you and you look at them at your time, at your own time. And then we have the process of urine formation. How urine is formed. Urine formation in mammalian. So